Please switch off or put your phone in silent mode so as to not disturb the student learning. Thank you. Assalamualaikum warahmatullahi wabarakatuh and a very good morning. Professor Dr. Zulfiqar bin Yassin, Director Institute of Oceanografi and Environment INOS, Associate Professor Dr. ID Muhammad Syawal and Muslim, Deputy Director Research and Services of INOS, Dr. Russell Rusty E. Brainard, Speaker for today's lecture, fellow lecturers and researchers, staff, students, ladies and gentlemen. Welcome to the INOS Research Lecture. Ladies and gentlemen, before we begin our event today, I would like to introduce our speaker. Dr. Russell Rusty E. Brainard is a Chief of Coral Reef Ecosystem Division of the National Oceanic and Atmospheric Administration, also known as NOAA, Hawaii. He will be giving a talk entitled Long-Term Monitoring of the Ecological Impacts of Ocean Acidification on Coral Reef Ecosystems. Without further ado, I would like to invite Dr. Rusty to give his lecture. Please welcome him with a round of applause.
Pagi, aloha. Thank you, Professor Zul and everybody for the very thoughtful invitation to come meet with you this week here at UMT. I very much look forward to meeting with all of you over the next couple of days, and thank you again for taking the time to meet with me this morning. As indicated, I'll be speaking about some of the work that we're doing to monitor the effects, the ecological effects of ocean acidification on coral reefs. There's a whole lot of authors shown here, and that's because what I'll be speaking about is really part of a global effort called the Global Ocean Acidification Observing Network. I'll be speaking about the components of that I lead primarily in the U.S. Pacific Islands, but we're also working with the Intergovernmental Oceanographic Commission, Western Pacific region, to establish a similar observing network here in the Western Pacific, and we're in discussions now with UMT and USM to establish similar stations that I'll be describing in and around Malaysia. Many of you already know, but this is just a global carbon budget. There's sources and sinks of carbon primarily through CO2. And as you can see, that from the emissions, there's sinks and there's also sources. Primarily the sources are fossil fuel emissions. The sinks are both increases in atmospheric CO2. There's some, which is here, and then there's also a significant sink of CO2 into the ocean. Here's projections under various scenarios of emissions of CO2. These are different IPCC models and this is through the end of the 21st century. There's some very kind of rosy projections where by 2030 or so we'll actually change our global patterns and reduce emissions. That's kind of the best case scenario, considered unlikely by most. The business as usual scenario calls for a lot more CO2 going into the atmosphere. And then this is some model output of trajectories of CO2. Actually, let me go back. That's actually an animation. And you can see from the pre-industrial, this is aragonite saturation state, which is one of the indicators of the ability of calcium or carbonate producing organisms, in this case particularly corals, to calcify their skeletons. And then this is marching through time. as we've been adding CO2 to the atmosphere, about 25% of that has been being absorbed into the ocean. And that's fundamentally changing and will continue to change the chemistry of the ocean and the ability of many of the organisms in the ocean to perform many of their vital functions. I'm going to change, and it's not just a theory, and that was a model projection, but we actually have observations. This is the famous Keeling Curve here at Mauna Loa Observatory, and that similar curve is observed all over the planet in the atmosphere. Station Aloha, an oceanographic mooring north of Oahu, is one of the best time slips. Is one of the best time series we have of changing, this is PCO2, so the partial pressure of CO2 into the ocean, and that parallels very similarly. So this is in the ocean, this is in the atmosphere, very parallel. This is computed pH, or measured pH at the station Aloha, and this is saturation state. So it's not just a theory, it's actually occurring. And there's a similar time series in Bermuda. Those are the two longest time series observations that we have of CO2 in the ocean. But again, it's not just a theory, it's happening, and it will continue to happen. And I'm going to change gears a little bit away from the science for just a moment, just to talk about why we care. And these a series of photographs from one village in Indonesia, and there's villages like this all over the world. Probably here, there's a fishing village, and the point is that people, you know, rich and poor, male and female, young and old, they depend on the ocean for many resources and their livelihoods in many cases. And they're providing many ecosystem goods and services to these people. So we're very dependent on our oceans, and our oceans right now are very much threatened by this absorption of CO2 into the ocean and the changing of the chemistry of the ocean, which is what we call ocean acidification. And that's because the pH is slowly lowering, and it's already decreased by about 30%. Kind of going back to the science, most of what we know about ocean acidification is from laboratory experiments and modeling experiments, and that's important. And those are foundational pieces of work, and in most cases, particularly for coral reefs, those controlled experiments kind of give us some scary news. Yeah, and these are experiments where they put a species of coral or crustless coral and algae or whatever the taxa of interest is into natural seawater at present day conditions, and then they put similar replicates of that, and then they change the seawater chemistry to what it will be in the future. So they're essentially adding CO2 to that water to simulate what will happen and to see the organism response. And for species after species after species, particularly your calcifying species and coral species and crustless coral and algae, those experiments have pretty much shown that corals' ability to calcify their skeletons or crustless coral and algae to produce that calcium carbonate is severely decreased. One of the more famous papers is by Langdon and Atkinson. Now it's fairly dated, but has been replicated numerous times. This was from the biosphere experiments, and essentially these are calcification rates with changing saturation state, and this is over time different CO2 levels in the atmosphere. There's different measures on here, but essentially the ability for corals to calcify continues to decrease as saturation state decreases, eventually actually reaching a threshold where not only does it not grow, it actually starts dissolving. And this is the trajectory we are with increasing CO2. So there's lots of reasons for concern for coral reefs. And then this has also been modeled in many cases, and there's a ton in the literature, so I've chosen a couple of examples. In the literature, this is an example of the percentage of reefs under different saturation states. I don't know if you can read that. These are saturation states. So this is pre-industrial up here. This is kind of now, this is, well, it depends on which emission scenario we're in, but this isn't too distant. We've now surpassed 400 parts per million in the atmosphere. And this is probably something what will be like near the end of the century. And what we'll see is that the large majority of reefs are down in this, in the future, are down in this very low saturation state condition, where in these response experiments, those corals aren't able to calcify and actually start dissolving. So again, this is all from controlled experiments and projections of that. What we have is very few observations in nature. We haven't documented this well. I've showed you the time series from Station Aloha. We have some decent observations in the open ocean, but even those only go back 20 or so years. We don't have long time series measurements, particularly of the biological response. And so what I'll be really talking about is integrated observations of the chemistry and the ecological response to acidification, specifically for coral reefs. I would advise doing that for other ecosystems as well, because it's not just coral reefs that are going to be affected by ocean acidification. Pelagic fisheries, tuna fisheries, all marine ecosystems are going to be affected in different ways. And right now we don't have we don't have adequate science to tell us exactly what's going to happen. And people often think, well, how, you know, it won't affect the tuna. Well, if you look at the food web of the tuna, it goes up, and kind of at the top of that food web or the bottom, depending on how you look at it, is much of your plankton, and much of your plankton is calcareous, also calcium carbonate forming. So we're fundamentally going to be changing the chemistry that forms the base of that food web. So it will also affect much of the food we eat in some ways. There's going to be winners and losers. So I don't want it to sound as though the ocean is just going to die. It's certainly not. There's going to be lots of life in the ocean. It's just going to be very likely different than what we've adapted our society to. From the kind of balance, and this is very simplified here, what we're trying to measure is the net production of calcium carbonate versus the net removal of calcium carbonate. And the removal is both the dissolution, but also the bioerosion. So we're actually trying to measure bioerosion and dissolution and calcium carbonate production in maintaining this balance. The other main thing that we're quite concerned about is just biodiversity in general in the ocean and on our planet. There's been 5 mass extinction events where we've lost greater than 50% of the diversity of life on our planet. More recent publications have shown that extinction rates have already increased over 100-fold. Losses of species, and the scary part with many of the things humans are doing, both in terms of climate change as well as overfishing and everything, the predictions are that we will increase extinction rates more than 100-fold this century as well. And what we don't know is what that will mean in terms of lost ecosystem services. Those things, again, that I showed, those pictures of people. What will people lose as we modify these marine ecosystems? There's much in the literature, particularly over the last decade, that we have already entered the 6th mass extinction event. That's kind of reflected in the fact that we've already increased these extinction rates 100-fold, and by the end of the century that will be 1,000 or 10,000-fold increases in extinction rates. So we're well on our way to being in our 6th extinction event. It's already been named, it's published in the literature as the Anthropocene, meaning that this is the first mass extinction event caused by humans. So it's a huge change. And from an ocean acidification perspective, there's some in the literature, and of course some of these events were 260 million years ago, so there's not complete understanding, but much of the information from the paleoclimate record suggests that most of these mass extinction events also co-occurred with increasing CO2 or ocean acidification events. And it's not to say that ocean acidification is what caused the mass extinction, but they coincided with each other. Speaking of biodiversity, in the ocean, this is a National Geographic cartoon from some data showing what we know about the diversity in the ocean is very, very limited. And this has several different papers that have come out. We just published a paper earlier this year, coming up with a new estimate of the number of species on coral reefs, and that has varied quite a bit. The new number is kind of a mean of around 830,000 species occupy coral reefs. That's multicellular species. The take-home message is most of the diversity on coral reefs we've not even documented yet. We don't know what functional role much of that diversity plays. There is evidence from many ecosystems, from rainforest to other systems, that this diversity helps buy these ecosystems resilience. If you lose some organisms, if there's others that are close in functional traits, then they may be able to be replaced. If you start the more diversity you lose, the more you start losing resilience of that ecosystem recover from perturbations. I mentioned earlier that under an acidified ocean, it's not all losers. There's actually going to be some winners. Some things do better with more CO2. We know that. Many of the plants thrive with CO2. They take in CO2. So it's not again, there's still going to be plenty of life in the ocean and on land as well. It just will be very different. So some will thrive. Some have adapted to dramatic fluctuations. They may be able to do better, and others are particularly vulnerable. And right now our evidence, again, from the response experiments in laboratories suggests that it's many of the calcifying organisms right now, to the knowledge that we have, are the ones that are probably the most vulnerable. And for the systems that I study, coral reefs, the reef building components, both corals and crustose coral and algae, the cement that holds reefs together, are particularly vulnerable. But again, that's all based on response experiments where they put one species in a tank and they change the chemistry of the water over a couple of hours to something that's going to happen over centuries. And that's really where our knowledge comes from. Nature may have much more resilience than those control experiments. We hope that. I mean, that's kind of because in a real system, there's these hundreds and thousands of species working synergistically with each other. It may be, and I actually put my money on the microbes. The microbes can evolve incredibly fast. They have generation times of days and hours, whereas the coral itself might not be able to adapt under the time scale or evolve in the time scale that these changes are occurring. But the holobiont itself, which includes the microbial community that it lives with, may be able to adapt to provide some of those corals some resilience, the ability to survive, the ability to continue calcifying even under these conditions if they can keep evolving. That's speculation at this point. We don't know that. But I think that's one of those things that we need to hold on to for hope. Otherwise, it kind of gets to be kind of a doom and gloom and kind of a too overly scary tale. I think the ocean has a lot of resilience. What we need to do is keep understanding that resilience, which is what you all are doing. You're studying. You're trying to understand how these systems work so we can better inform society. Again, kind of on the why it matters, many of the ocean issues keep coming back to fisheries because that's a close connection to people. People eat fish, people earn their living and livelihoods from fishing. So just an example from some of our Australian colleagues of why losing habitat. In this case, it was a coral bleaching event from some studies where there was mass bleaching and what happened when there was a mass coral mortality event. And in that particular series of studies, 10% of the reef fish declined because they were directly dependent on the coral itself. That's actually what they fed on, is the coral. But it ended out 75% of the species, the fish species, declined following that mortality event. And 50% of the species, and kind of the cartoon shows that relationship, and this was actually provided by Phil Mundy, a colleague from James Cook University. So this is kind of time now in years of a mass coral mortality event. In this case, it was a bleaching event, shown kind of here, from doing well to mass bleaching to dying and then collapse. So initially, the coral cover went down because the coral died. The macroalgae went up because there was new real estate, and macroalgae settles rapidly. So macroalgae went up, but the complexity, the structure of the reef was there for many years until, and sometimes it will recover. In this particular case, it didn't recover, and we actually lost some of that complexity. For the fish, there was that 10% so decrease initially, the ones that actually feed on the coral, but most of the reef fish did okay for a while until that habitat was lost. They lost that three-dimensional habitat complexity or structure, and then we started losing those fishes as well. And that's just an example again. The fish weren't directly connected to that warming event. Same with ocean acidification. Many of the fish, so far from what we know, which again is very limited, the fish have more capability in many of the fish, and again we don't know about many of their life history stages, so there may be very vulnerable life history stages, but they may be able to survive the actual change in the chemistry of the water, or temperature in some cases. But these complex habitat relationships of the other things that they do depend on may be very vulnerable to acidification or warming or some of these other global changes. So now I'm going to start talking a little bit about the observations. The observations that I'll be talking about are part of NOAA's National Coral Reef Monitoring Plan, which is a national plan. I lead the portions in the Pacific, which is actually, we've been running since 2000 and 2001. Most of the plan is kind of a typical coral reef monitoring program, and I'll show some of that because I wouldn't propose to just monitor for acidification in absence, is to connect your ocean acidification monitoring to your coral reef monitoring, because that's what people really care about, is the fish and the corals and things like that. What we did is we added additional things on top of an existing long-term coral reef monitoring program, which I'll talk mostly about that, but I'll share with you what we're doing as our basic coral reef monitoring. So that program is the Pacific Reef Assessment and Monitoring Program. The area where we work is all of the U.S. areas of the Pacific, and it covers a vast, vast region, from the populated main Hawaiian Islands to the northwestern Hawaiian Islands, all the way to the territory of Guam and the Commonwealth of the Northern Marianas in the Western Pacific, down to American Samoa in the South Pacific, and then there's 7 remote islands and atolls in the Central Pacific. We, as a typical monitoring program, we do long-term monitoring of abundance and diversity and condition and distribution of the fishes and the coral and the macroalgae and the macroinvertebrates and the algae. Then we added this component to look at those additional things that are affected by acidification, and we're still trying to improve our social economic monitoring. The approach that we've taken, because of the vastness of the area, there's many ways. There's a lot of discussion about is whether we should choose a few places and study everything, kind of sentinel sites. Because of our legislation that creates our funding, we're actually tasked with monitoring our nation's reefs. So we've chosen to do this wide but thin approach. So we're actually monitoring all of our nation's reefs, in this case in the Pacific Islands, using consistent, repeatable comparisons. So we're using the same methods at all of these island and atoll systems across the Pacific. And we're using a stratified random survey design to maximize our statistical power to detect change at an island scale. We're not focusing at a site scale. All of these most of these are very small. Many of them are uninhabited islands. Our unit of change is statistically sampling that place enough that we can detect change at the island scale. For some management purposes, that's not enough. They want to know how a particular watershed, reefs off a watershed are doing. Our program isn't adequate. It could tell how that particular watershed is doing compared to the rest of that island, if that were the example. Because of the remoteness of these areas, again, thousands of miles in some cases from populated areas, we can only visit these areas on a 3-year cycle. So I have a team right there actually they arrived today at Jarvis Island in the Central Pacific. They've been gone the last 2.5 months on the way down from Hawaii to Johnston, Helen Baker. They spent a month and a half, almost 2 months in American Samoa. Now they're on their way back. So it takes us about a little over 3 months every year of sea time aboard a fairly large ocean research vessel that can support 22 scientists to do these surveys. Again, part of that is just because they're very remote. There aren't any other ways, there aren't any airstrips, there isn't any civilization in most of these locations. So we're kind of trapped, in our case, to a research vessel as our primary environment. So all of our areas, just an example of what we do. First, we map all of the areas, doing multi-beam to get depth in the deeper waters, in the very shallow waters of the coral reef. We use satellite altimetry and color imagery, and then for land, we do digital elevation models. We combine those and interpolate to get a complete model from the top of a mountain down to the bottom of the reef. And then we classify the habitats and stratify it and then subsample. So this is an example of the different layers that we produce of our habitat. And then we randomly, based on different stratification, we randomly select points, and those are regenerated each time, so we don't revisit the same sites every 3 years. We re-stratify and subsample those strata and do our benthic and fish monitoring from that. And as just an example, not to go too far, from those stratified random, this is from the fish surveys, we can actually come up with estimates of abundance. Actually, this is by island in American Samoa in this particular example, but we can do it actually not only by families, but by species, dependent on the abundance of a particular species. So this is actually informing our stock assessment from our basic reef surveys. This paper actually just came out a couple weeks ago, kind of showing, by Ivor Williams, showing kind of plot, and that was in PLOS 1, showing the distribution. This is islands across the Pacific that we survey. The ones on this side are remote, uninhabited islands. The one on that side are populated islands. The colors on here are the fish biomass for that island in these kind of consumer guilds or functional roles of the fishes. The gray in these figures is the computed biomass based on a kind of a generalized additive model of looking at all of the relationships from a broad, mainly the environmental data, which the kind of the biggest drivers in determining fish biomass from a very large data set were human population presence. You can see here, these are the populated islands, and oceanic productivity. So some areas that we survey have very high oceanic productivity, and I'll show that visually separately. So they can actually support, whether there's fishing or not, they can actually support higher population. So the gray bars are based on the projections of what that system is capable of supporting. And then the blue is what we're actually observing. And you see in the unpopulated areas where there's not fishing, the biomass that we observe is very similar to the biomass that's predicted. Some reductions in some areas, but you compare that to the areas where there's people. So these gray areas are predicted based on what that system can produce from an oceanic productivity sense. And these are what we observe. So the difference between these gray and what's observed is the removal from fishing, primarily. And a couple of examples are shown here. One from Swains Island, where it's a very small island in American Samoa that has less than 10 people living on it, essentially a family. And there's about a 20% reduction from what that system could support without the presence of people. And then there's a larger, more populated island also in American Samoa, Tutuila, which has a lot more people, and that reduction is about 56% of the biomass has been removed from what the system should be able to produce. Another way of showing this, this is the fish estimates by functional group. This is the piscivores, the secondary consumers, primary. And what you see is in the larger fish, and we also can plot this by size. Again, this is unpopulated. The red areas are the populated areas there. And we can see what we're particularly removing is the piscivores, the larger fishes that we target as fisheries. And where in many cases we dramatically removed the biomass of those fishery targets. In a spatial map sense, it's the same data shown, and here you can see this is just the piscivore biomass. The populated areas on here are these kind of purplish or pinkish colored areas, and you can see that the biomass, so the main Hawaiian Islands, the Southern Marianas, which is where Guam and Saipan are, and Tutuila and American Samoa. The remote areas have healthy populations of the reef fish. Yet another tool, so that's what we do from our stratified random surveys. Another approach that we use to get much more spatial coverage, and a better tool that we found for looking at certain of the fishes that are affected, particularly the large fishes, the sharks and the jacks and sometimes the snappers, are very much influenced by divers. Sometimes they're attracted to divers, and others they stay away from divers, so they don't show up adequately in surveys. Our towed diver surveys are continually moving, and we find that for the large predator populations, we get much better estimates of abundance from those. And those also, we use these towed diver surveys to get much broader spatial coverage of our benthic surveys, and it's really the only method we have for getting good estimates of abundance for certain of our conspicuous macro invertebrates like crown of thorns that have significant reef effects. And from that, we can actually, we don't have the resources to analyze all of the images. We're taking on this tow board, we have a camera mounted on the bottom that's taking a photograph every 15 seconds. So we have a permanent record for all of the years around everyone. So this is, as an example, Palmyra Atoll. So every island that I've shown on that larger map, we have a complete circumnavigation of every one of them where we're doing these fish and benthic surveys. And for every one of those, we have a photograph every 15 seconds of the benthos. And for one of the PhD projects, they actually analyzed all of those images. And right now, we only analyze routinely about 1 out of every 100 image to get estimates of benthic cover at the island scale for change. But we can see there's tremendous structure. This is coral cover, this is crustose coral, these functional groups that were shown in this. We can classify the imagery to functional groups, and then we can actually, around an entire island, we can actually look at how the benthos changes spatially. And then this is kind of combining some of the different types of information I've shown. So the size of these pie charts is the abundance of fish. The colors of the pie charts are the benthic composition, and the map in the background is chlorophyll. And what you see is some of these areas, you have very high fish biomass. These are that's both because there's no fishing, but also because there's tremendous productivity. That's equatorial upwelling, and there's also, where they're at today, at Jarvis, intense topographic upwelling. And so that's also affecting the reef. So you have the pink, the red is coral cover, and the pink is crustose coral. And so those are very, very productive, healthy reefs. Some of the northern areas of the northwestern Hawaiian Islands, some of the areas in the northern Marianas are also biologically have very healthy fish biomass as well as benthic production because of upwelling. Watching my time, we're also doing disease and condition of coral surveys. I won't go into this in any detail, just other than to let you know that those are there. So on top of that kind of core coral reef monitoring, we've added components to help us. What we're trying to do is create something like the Keeling Curve, but for both the chemistry in the seawater of coral reefs at each of these locations, and then the key indicators that we expect to change from ocean acidification. In addition to what we we're already measuring are those large macro things like fish and measures of benthic composition. We've added measurements of the seawater chemistry, the carbonate chemistry, at all of the islands where we work. We've started, because of the response experiments have given us this doom and gloom story, we're actually measuring at these locations calcification rates, and we're doing multiple different approaches for that. We're also measuring bioerosion, that plot I showed earlier, trying to reach this balance between production and removal of calcium carbonate from the system. So that's why we're doing these components. And then I'd also mention one of the other components is biodiversity. Well, most of what we know about biodiversity on coral reefs is the diversity of fish and the diversity of coral. Together, those taxa represent less than 1% of the diversity of a reef. That 830,000 species on reefs, there's less than 1,000 species of coral and a few thousand species of fish. The rest is mostly organisms that live within the reef matrix, mostly small cryptic organisms, and most monitoring programs, including what I've described so far, don't monitor any of that diversity. So as part of the Census of Marine Life project from 2005 to 2010, we developed a tool for monitoring that cryptobiodiversity. That's called an autonomous reef monitoring structure. So we've added that to as one of those things that we're trying to measure across space and time, as well as the microbial diversity. One of the things I mentioned earlier is the microbes may be the thing that actually save the day and make things not as bad as what we think they are when we put things in a simple, very simple experiment, because again, they can evolve. So we've started, at least in a simplified manner, at least measuring some components of the microbial community routinely. So I'll describe just a little bit about how we're actually doing that. We can't do all of those things at all of the locations. So we've gone to a hierarchical design of having three kind of classes of stations, where the class 3 stations, we try to do everything, but we only have 3 of those across the vastness of the Pacific. So there's one there, and there, and actually we so far don't even have, that one's not even there yet, and that one's not even there yet, because we don't have the funding for it. Because to do everything, again, the Sentinel site sort of stuff takes more money, and we don't have more money. So many places where all these yellow dots, at 4 locations within each archipelago, we're doing what we call class 2 sites. And in all of the places, in class 0, class 1, we do the water chemistry. Class 2, we do a lot of more of the biological response. In class 3, we have buoys to actually do kind of continuous measurements of the chemistry, and I'll show you some examples of that. And we're on this same kind of 3-month or 3-year cycle, 3 months at a time. So at the 3 class 3 sites, we're planning to have continuous measurements of PCO2. Right now in Hawaii, we have a more autonomous PCO2 sensor, buoys like this that every 15 minutes are measuring the CO2 in the seawater and some other oceanographic parameters. We're trying to establish something similar next year in American Samoa and the following year in the Marianas. And then what those provide us is this high frequency variability of the carbonate chemistry. We know there's seasonal cycles of the variability that's shown in the data. There's also significant diurnal variability of the carbonate chemistry. During the daytime, these systems are mostly photosynthesizing, so they're taking up CO2, and at night they're respiring, so there's very strong diurnal cycles. So while we can't measure that all the time everywhere, we're trying to get some measures of it at some places. We have a much larger number of class 2 sites. We have 52 class 2 sites. So that's what I'm going to focus on next again right now. And everything in a class 2 site is also at a class 3 site. The difference is they don't have the buoy. They don't have the continuous chemistry being done. So going into a class 2, at a class 2 site, again, we have 4 per archipelago, also driven by cost. And so at a class 2 island, we actually have 4 stations. Well, one, we do water chemistry, also stratified random collections of water for the chemistry. So that's where we actually are measuring dissolved inorganic carbon and total alkalinity. And from that we compute pH and we compute saturation state. We do upstream and downstream and on the reef chemistry, so we can actually in net ecosystem production. And that's as we get offshore water, as that flows over the reef, it's interacting with the reef itself, where the reef is, if it's calcifying, is taking in some of that CO2. So it's changing it, so when we measure the water after it's crossed the reef, its chemistry has changed. So we're actually getting calculations of that. And then in addition to the water chemistry and the upstream, downstream, we then have 4 sites at these particular the subset of islands, 4 per archipelago, where we put permanent stations in, where we deploy a bunch of instruments. And I'll show you, I'll then zoom in to what's at one of those sites. So now zooming in to now, at 4 sides of a particular island, we have 4 class 2 sites, and there, and you can see the dimensions are 5 meters by 10 meters, so now it's small in space. And in every one of those, we put a precision temperature recorder. We put 3 of these autonomous reef monitoring structures, the arms. We put 5 of the calcification accretion units, and they're to measure the production of calcium carbonate, or accretion of calcium carbonate. We put 5 bioerosion measurement units, and that's again to get a measure of this bioerosion. We do water sampling both at the reef, right at the reef substrate, as well as at the surface at all of these sites. And then in a subset, the cruise that's out there right now, we've acquired some CFAT pH sensors. So at a subset of sites, we're getting continuous measurements only for a couple days, because we're only there a few days. So we can start getting the diurnal cycle of pH. We also have some things that we call pucks, which are portable underwater collectors that were designed by Chris Langdon in Miami. And those actually we deploy a number of them, and every 3 to 4 hours they actually collect water for us so that we can get DIC. So again, we can only do that for 1 or 2 diurnal cycles, but that's to help us characterize that diurnal cycle that we're otherwise missing of the chemistry. And then a subset of sites, we also do coral cores, and that gives us a rate of calcification or extension of the massive reef-building corals. And then we do, well, mostly we do photoquadrats along the boundaries of this climate station. At a subset of sites where we have the resources, and in the northwestern Hawaiian Islands we had additional resources, we actually did photomosaics of the adjacent 5 by 10 meter slot. So we can revisit that every 10 years and see actual changes of individual colonies of that entire space. Just as an example, if this works, I'll show you kind of the installation of a climate, at one of our climate stations. This was from last fall in the Marianas, or actually last spring in the Marianas. While it looks like our divers swim really, really fast, it's actually sped up. So this is about a 40-second snapshot. But all of what the point of this is everything that I just showed there, which seems like a lot, it's done in a dive or 2. So they're doing one of those sites every day. So the places where we have the 4 sites, they're doing a site a day. So it may sound like it's a lot, but it's very achievable. And you don't have to be super speed. You can do it at normal speed. And we're really constrained being on a research vessel where we don't have much time in a place. If you're at a research station, you could do some of these one day, something else another day. You could actually do the same people. So we're having to be very fast and efficient because ship time is very, very expensive, and we're piggybacking on another operation. So in kind of summary, we go from kind of our class 3, which have this also, to these other components, which are in our class 3. That's what an arm looks like. That's what a BMU looks like. That's what a cow looks like. This is the coral coring, and this is some of the probes for the CFAT sensors and some of the other oceanographic sensors that we have. So just quickly some kind of preliminary results. This is pH these are the actual measurements of computations of pH. And what we can see is some areas are more acidic across the Pacific Islands where we work. Some areas are less acidic. Mostly this is because of upwelling patterns. And then there's another way of plotting this on a larger scale, including we also have been working as part of the Coral Triangle Initiative. So we have a bunch of surveys around Timor-Leste, some surveys in Papua New Guinea, and actually I'm not sure why it's not on this figure, also some in the Philippines. And what we're hoping is we also have some soon, or you already have some in Malaysia, and again, more broadly around the region. And just to show, so when they, on this earlier figure, let me go back one, the background color is the open ocean conditions from these large global projects. So those are kind of mean conditions. And you can see on the reef, it's roughly similar to what the broader oceanic conditions are, which is good news. But we also see that if you looked at any one of those spots, there's actually significant variability around a particular island, whereas this is actually showing the mean of all of those, and that mean of all of those is somewhat similar to the oceanic conditions. But there's certainly places that have different carbonate chemistry. And this is just the measurements for the areas where we've surveyed in the Coral Triangle. And again, we hope to expand this broadly through a collaboration with IOC and with Westpac. Kind of statistically, we have a fairly large N, well over 1,000 water samples. The cruise that's out there now is doing another 400 water samples. So we're slowly, rapidly getting an abundance. So these are paired for the ones on the left in the colors of the measurements on the reef. These are the measurements at the surface. And in some cases, they're very similar with more variability at the surface. And so this is the American Samoa. These two are the Marianas. These are the Coral Triangle. And we also see some spatial patterns where some areas are naturally, and this is in aragonite the computation of aragonite saturation state. And you can see some areas are fairly tight with very low variance, and others are very high variance. Well, they, again, could be just diurnal cycle effects. Or other things that we don't understand yet. This is kind of our first kind of attempts at looking at some of the temporal change. So from some of the equatorial islands, so these here are right on the equator, that's the equator there, it passes right through these islands. And what we see is very strong El Nino, La Nina signals. So in 2010 was a moderate El Nino event in this region of the Pacific. And what we see then, and this is these two islands in red, the yellow island, and the 2010 during El Nino, you have very high saturation states. And that's because upwelling during El Nino, you have warm water sitting on top of preventing upwelling, essentially. And there you have very high saturation states. So that was actually conducive for coral development. The opposite condition was a La Nina condition, which is very strong upwelling in these areas, bringing up lower CO2 waters. And sure enough, we're actually seeing that in our measurements. We have these significant, fairly precipitous drops during that La Nina condition where upwelling has been suppressed. So again, as we get a longer time series, we actually what we're really looking for is more of a Keeling curve of CO2. But in the short term, you know, we're just in particular because we're only measuring every 3 years. It's going to be a long time before we actually can measure that Keeling curve. But that's really the primary goal, is having that definitive data set 30 years from now of whether pH, saturation state, DIC changed in the ocean over these long periods of time. People usually use these DIC total alkalinity plots at a specific site to see a diurnal cycle. They look at the diurnal cycle, and that's where they see that during photosynthesis or respiration, you end out on different places of this. We're actually using this differently to plotting all of ours in less in a temporal sense and more in a spatial sense. And we can see some areas are more aimed at dissolution, other areas more at calcium carbonate production. I'll skip through a couple of these. So that's then an island. And then if we take, so that just included all of the samples to then just regions. And this is kind of the statistics. And that then shows us that the Pacific remote islands and the main Hawaiian Islands are more, right now, from the surveys we have, more on the dissolution end of that. Other areas, like the Coral Triangle, are more on the formation of calcium carbonate. Briefly, the coral coring we're doing with Woods Hole and Ann Cohen's lab at Woods Hole. And one of the things that was a very significant surprise from what we expected, again, from the laboratory experiments, we expected under low saturation state we'd have lower calcification. And what we're actually measuring is some differences to that. And what we found is in some areas where you have strong upwelling or high nutrients, even with low saturation state, corals are still able to calcify well if they're being fed enough. And now that's been replicated in laboratories, so when we actually started the collections, that wasn't the hypothesis. Likewise, we've sampled those cores, which are CAT scanned for bioerosion, and what we see is bioerosion is higher at low saturation state. This is saturation state here. But one of the things that really surprises us, for high nutrient reefs, the sensitivity to saturation state is much, much greater than for low nutrient conditions. This is just what we're doing with the calcification accretion units. This is what it looks like at deployment. We co-locate with one of the bioerosion measurement units, which is just a piece of calcium carbonate that we CAT scan before and after to look at change. So this is what a cow looks like upon recovery. After a couple years, we dry them and then essentially weigh them, and then we get these spatial distributions for each of the cow sites across the Pacific. Actually, Nicole Price, a collaborator at Scripps, published the first of the cows manuscripts in 2012. Just learned this morning that a manuscript from this data got accepted by PLOS One today. But essentially, what we're starting to do is document for the first time what current rates of calcium carbonate production are. From the BMUs, we're going to have, and we started putting those out later. We're just now getting our first ones back on the cruise that's out there now. We'll also then have spatial maps of bioerosion. And again, what we're really looking for is doing that every 3 years, so that in 10 years, in 20 years, and 30 years, we're actually seeing if ocean acidification is having those long-term changes. And then this, probably hard to see, but this is something that's just been plotted up actually since that paper was submitted, where we actually have for the first time across the Pacific, this is the net production of calcium carbonate from the calcification plates. And I don't know if you can even see those dots, but they're quite small. There's a dot there, there's the Hawaiian dots, and some of these are fairly small, versus these which are fairly large. So this is net accretion of calcium carbonate. We can overlay the measured aragonite saturation state, and it's actually kind of the story you would expect is the areas with low saturation state have low calcium carbonate production. The areas with high saturation state are showing high production of calcium carbonate, which is what the theory would, most of the time you find something completely different than what the theory suggests. In this case, it looks like theory is holding. And actually, again, those were means. There's actually, if you look at specific islands, it's much more complex. And again, why having things on multiple sides of islands helps give us a better understanding. So in these cases, some areas like Rose Atoll is kind of similar around the entire forereef. Other areas, one side of the island is very different than the other side. And again, we think that's because, and this also agrees with what our series, the upwelling sides of these islands is this side. When it's upwelling, it's bringing high CO2 water, low saturation state on this side, high saturation on that side, and that's where we're having the production. Finally, going to the autonomous reef monitoring structure. So this is what an arm looks like upon recovery. This was from an area with very high crustless coral and algae. But there, a tool, this is a graphic from Smithsonian, that David Litchwater made of the diversity in 1 cubic foot of coral reef from the Maria Biocode project. And that's very similar to the size of an arm. They're only this big. And I'll show you what is kind of, this is what we have deployed around the globe. So in addition to what we have, we've also, with partnerships, been deploying them all around the globe, including around the European seas now. And this is what they actually look like. They, in a couple videos here, of putting them together. Also, he's very fast. It's all about getting efficient. And then just, we stake them to the bottom with weights, and I'll skip ahead a little bit. So this was some deployed in Maria. And then upon recovery, we leave them out for 3 years because that's what our survey cycle is. So this has a mesh net on the inside of it. We cover the arm and strap it down to the arm so that none of the organisms can escape, because it's not just the sessile, it's both also the motile organism. And then we bring it back to the surface and analyze it. So we take it apart, we photograph the top and bottom of each of the 9 layers. You can see the various different functional groups on each of those photographs and analyze those. But the primary tool that we're trying to get at, and the way that we actually get at so much of the diversity, is by sorting it into different size fractions. So these are the large organisms, those greater than 2 millimeters. So essentially the ones that you can actually see and you can identify. And we train these guys to work really fast too, to sort. And essentially that's from an arm. They're organizing and sorting all of those, and then they barcode and identify those organisms. And this is kind of what they were sorting, is the various crabs and snails, and again, tremendous diversity of organisms on these reefs that settle into these arms. And then the other component, and then after we take voucher specimens and barcode individual specimens, everything else gets scraped. All the sessile organisms get scraped together. And as this video shows, we actually blend them, turn them into smoothies in case you're hungry. Smoothies after the talk. Looks delicious, right? And then we mass parallel sequence this. So we do next generation sequencing of that. So we subsample that. And then what we get, and this is what's really surprising, is again, an arm is only yay big. The numbers of different types of DNA on an arm are almost always well over 1,000, up to 2,000. So those that each represent, I mean, right now we don't know what most of those are, because we just have the DNA sequences of them. Most of them have never been identified. So they're operational taxonomic units in kind of genetics parlance. But again, somewhere between 1,000 and 2,000 species per arm across a whole bunch of different phyla. What are there, 33 phyla on the planet? In an arm, you'll have 18 or 20 phyla represented in that small part of real estate on a coral reef. And you can see spatial patterns from different areas of the Pacific. You probably can't read that, but this is in the coral triangle where we're seeing much higher diversity. And just, I think this is my last slide. We're still finishing kind of the developing, just finalizing the development of the genetics method. So we don't have much yet in terms of those results other than what I just presented, and some colleagues, Nancy Knowlton and Matthew Luray, just published kind of a methods paper on the genetic part of it a couple of months ago. But from the taxonomic side, this is Coral Triangle, the Marianas, the Pacific Remote Islands, main Hawaiian Islands, going this way, and at the alpha, or the unit, the arms level, and then again, this is taxonomy, not genomics. And genomics, we think we'll get a very different picture. But not surprisingly, the coral triangle at the gamma diversity level, which gamma is kind of at the landscape level. If you average kind of all of them, the unit level is an individual arms. And this is a little bit surprising to us. We don't know if that's because of this is actually taxonomy and we didn't know enough of the species in these areas. We actually think that when we do the genetics the coral triangle, it will be more similar to this as being highest in the Western Pacific, but that's still to be determined. That's actually some fairly new results. We're now kind of going into production mode for the genetics over the next couple of years. I think you'll see some pretty amazing and probably some surprising things that we won't know why that is until we think about it a lot more. So with that, I guess I hope I left you with that ocean acidification will increasingly impact coral reefs, diversity, fisheries, and those ecosystem goods. We need more. We need to continue response experiments and modeling, but we need, in various areas around the globe, consistent long-term observations of the chemistry and the biology on coral reefs. We hope Malaysia becomes a partner from all discussions we've had so far. I think UMT will be one. I think USM may be a partner in this, and I look forward to discussing with all of you how we can all work together to get more observations from Malaysia and from the Western Pacific region. So thank you very much.